So I think we're moving along here to Professor Ross, Andrew Ross from Oregon State University, and I asked him to take on a pretty difficult task. As we were talking about wheat and breads and all these things, there are a significant amount of people that cannot eat these things. But then there's also some individuals that are excluding gluten and FODMAPs probably for the wrong reasons. And so I asked Professor Ross to address this. What's real about gluten and FODMAPs and what's not? Thank you so much for joining us and preparing for this.
Okay, what's real and what's not? So let's, I'm going to just diminish the expectations here a little bit, or what's the truth about them. It's basically what I'm going to try and present in the next 20 minutes, and it's a really big topic and it's hard to summarize it in a short period of time, is my interpretation of recent observations on this. And some of the things that we'll talk about are circular in terms of referring to the work of Marko Gabetti and the Finnish group, Kaisa Paltinen and Mayuka's group. And so I'll get to cut to the chase here. Okay, I have no idea what you're looking at. Hopefully you're looking at my smiling face with a bunch of 100% whole wheat spelt.
We've got it, it's looking good.
Excellent, okay. So I'm just gonna talk about gluten and FODMAPs in the context of grains. That's kind of, I think, where I ended up going with this. So just an aside, this was more of the picture that Kaisa Paltinen showed us this morning. Other than high resolution pictures of the surface of Mars, this is probably my favorite picture in all of science. It just is so evocative of what's going on in the difference between these 4 cereal species in terms of the distribution of fiber. I also wanted to reflect on the fact that Michael Gensler, I think a long time collaborator of Marko Gabetti's, has also shown the production of ACE2 inhibitor peptides in sourdough fermentations, which was that topic was brought up by Jan Lam earlier. Okay, so I'm going to talk about wheat-related maladies. I was going to make a joke and call them worms, but you know, I don't know that this is a joking topic. There are argued increases in what these maladies and the argued differences in what might be causing them. What a very brief sort of summary of what gluten is, what FODMAPs are, and why they can be a problem, and then some of these things have been floated as the cause of this, and one of them particularly is the wheat changing. Is it processing? And then I'm going to look a little bit later at wheat-related maladies and gut permeability, and that has come up again and again, the gut permeability issue. So the wheat-related maladies, we're familiar with celiac disease. This is an autoimmune disease. When I was monitoring the first forum, there was some questions and answers in the Q&A or the chat, and someone said, when someone has a severe wheat allergy like celiac disease is an autoimmune disorder, it's not an allergy, and the immunological signature is quite different. Anyway, it requires at least 2 things, and some argue that it requires 3. It requires a gluten trigger, it requires a genetic predisposition on the human side of it, which is risk alleles or gene variants for this human leukocyte antigen, and that's about 1/3 of the population. And then some argue a previous or an existing bacterial or viral infection. And each of these, with maybe the caveat that there's argument about the third one, are necessary but not sufficient alone for the development of celiac disease. There are a group of true allergies, Baker's asthma, dermatitis, hives, and frighteningly anaphylaxis, wheat-dependent exercise-induced anaphylaxis. And I get a bit annoyed with this one when I read the wheat-based literature that folks would like to sort of express to us that it seems that wheat is evil here and it's the only one that produces this exercise-induced anaphylaxis. In fact, the wheat-induced version is just a subset of the food-induced versions. So more than wheat as a food can induce this in susceptible individuals, and about 50% of all exercise-induced anaphylaxis does not have a food trigger at all. So it's not just wheat. And then, of course, there's wheat sensitivity, which I'm differentiating as not related to gluten, and then non-celiac gluten sensitivity. And the usual suspects. So as Steve Jones just talked to us about, the glutenins and the gliadins from the endosperm of wheat and their analogues in rye and barley. And then some non-gluten proteins like lipid transfer proteins, amylase trypsin inhibitors, and then a non-protein actor, which are these fructans, which is one of the FODMAPs. And wheat is one, particularly whole wheat, is one of the richest sources of that in a North American diet. So the argued increases, what might be causing them, that there's more gluten in wheat in modern wheats, that the gluten's different, that there are more FODMAPs, that there's not enough or not at all processing. And then, I'm going to talk about gut dysbiosis as a sort of overarching theme in this. Okay, so gluten, and as I said, the analogues, and I'm just going to say that, most of us are familiar with the idea that gluten is this viscoelastic proteinaceous mass that when we add water and energy to flour and we create a dough that allows wheat flour doughs to uniquely stick together and have these kind of elastic behaviors. It could take me 20 minutes if I was just going to go into to just explain gluten, and I need to just be very quick about it. These glutenins and gliadins are rich in these two amino acids, proline and glutamine. And this is a 7 amino acid long section of a gliadin. It's a gliadin peptide. And there are two things I want you to look at here. The first is the prolines. They have this ring structure, which creates a sort of kink in the protein backbone, which makes it difficult for standard proteases to get in there and cleave it. Hence the fact that these gliadin peptides are a little recalcitrant to digestion and may reach the small intestine intact or partially intact. The other is the glutamines, and I've circled in red here the amine or nitrogen containing group that sticks out on the side chain. And when these get into the gut, these amines can be cleaved off by an enzyme that's produced in the gut epithelium. This enzyme is called tissue transglutaminase, and it changes the glutamines to glutamates. And if you happen to have the risk alleles for celiac disease, the HLA risk allele means that this major histocompatibility complex is a good fit for the deamidated version. And if the deamidation doesn't happen, it's a poor fit. And if you don't have the risk alleles, this is not going to happen. And this initiates the cascade, which leads to the autoimmune damage caused by celiac disease. And I'm not going to go any further. I just wanted the gluten-specific part of it. The other part of this is the infection part. Why is this particular structure a good fit for this deamidated glutamate? And so one of the arguments that suggests the need for a prior or concurrent infection with the gluten challenge is that there seem to be, on some viruses and some bacteria, peptides that are very, very similar to this deamidated gliadin peptide. And so these are pre-loaded to recognize these gliadin peptides as a threat. So that's really kind of interesting and maybe tangential to what we need to know. This is what happens to the poor celiac sufferers. And this is specifically related to the structure amino acid sequence of gliadins, which is then prefaced on its richness in proline and glutamine. The FODMAPs, okay, I could take 20 minutes just describing what they are to you. So I'm going to talk about this in sequence across the top here. This acronym is for fermentable, oligo, di, and non-saccharides, and polyols. Polyols you might not be familiar with, things like sorbitol and xylitol. If you've ever read the packaging on sugar-free gum, you'll have seen one of these But fermentable means that they're reaching the large intestine intact, which means that they are able to be fermented. And in healthy individuals, this is arguably not a problem. So I'm going to talk about, these are the oligosaccharides of FODMAPs. So this one is a fructo-oligosaccharide or fructan. And these linkages here, we are unable to break them down with our own enzymes, any of us. And this always reaches the large intestine as a fermentation substrate. And for many of us, this is a very, very valuable prebiotic, which is, encourages the proliferation of butyrate-producing bacteria. These are galacto-oligosaccharides. We've heard about raffinose earlier today from Michael Gobetti in beans, but raffinose and stachyose in beans, but also raffinose in cereal grains. The disaccharide that's referred to is lactose, and not all of us are lactose intolerant. And so for some of us, this doesn't reach it's absorbed before it reaches the large intestine. Not all of us, this is the monosaccharide, have fructose malabsorption problems. And so for many of us, this is already absorbed and is not fermented, but for a bunch of the population, it does reach the large intestine as a fermentation substrate. And then here is a polyol. So you can see this alcohol group here. I've just made this is sorbitol, it's derivative of glucose. That's all you need to know about that right now. And we won't focus on polyols, except to say is that, yes, they can be synthetically synthesized or chemically synthesized, but if you're on a low FODMAP diet, you can't eat apples because they have sorbitol in them, naturally occurring. Okay, so gluten sensitivity and celiac disease, one of the arguments is that the gluten is changing in modern versus old wheats, and particularly that the gluten content of the wheats is increasing. And so I have a lot of converging lines of evidence that say this is not true, but all of that is mostly predicated on measurements of grain nitrogen, which also includes non-gluten proteins and non-protein nitrogen sources. And so this paper from 2021 by Pronin et al. Actually applied the ELISA tests that are used for detecting gluten in foods to grains, 5 varieties per decade from 1891 to 2010. And there's no significant difference between 1891 and 2010. If you look at the letters here, they overlap. So this has got B and C, so is this. You could dream up a downward trend here for this other ELISA test. It's flat. So there's no evidence there that the gluten, specifically the gluten content, is increasing. So I argue that that's not a cause. Gluten sensitivity is the wheat changing, in other words, in the composition of the proteins rather than the abundance of the proteins. And there's a particular class of gliadin called alpha gliadin, and within that class of gliadin, there's a 33 amino acid long sequence called the 33mer, which is reputedly the most reactive for celiacs or for folks who are at risk of becoming celiac. And so Pronin et al. Tracked the incidence of this across these, I think it's 60 varieties, I think 120 varieties, I think there's 12 decades here, but more than 100 varieties. And there is no evidence that the incidence of this 33mer in this group of wheats was increasing over that time period. And so the celiac reactivity hasn't increased. And this is their wording up here. They identified both old and modern cultivars containing both high and low contents of these CD-active peptides. There is argument in the literature about this. Hetty Vandenbroek in, I think, somewhere in the 2010s, the 20 aughts, published that said that there was an increased incidence of this in modern wheats, the particular 33mer, but I have a grievous disagreement about the way they selected the wheats that they chose. And so, you know, this is not all settled. So looking at it in a kind of more general sense of celiac reactivity, this is a review from late in 2020 by Di Suter and Frank Bekesh. And one of the things they looked at a meta-analysis of values reported in the literature, and really there is very, very little difference, certainly between durum, spelt, and these, I would say, modern wheats. I make no distinction between heritage and modern wheats in terms of celiac reactivity. One could, with your imaginative, suspect that Emma is a little bit less reactive, and that, but this is repeated in the literature that einkorn definitely has less reactive, is less reactive to celiacs, but it's still wheat. It still has celiac reactive epitopes, and it's still not safe. There is other evidence, particularly for Emma and durum, and that would include Khorasan as well, these tetraploid wheats, that because they don't contain the same, so I don't want to get into the genetics because that's going to take an hour. This contains the A genome alone. These contain two subgenomes, the A and B, and these three contain three subgenomes, A, B, and D. And the D genome is the one that is coding for the most reactive celiac epitopes. And so there is some evidence that these are less reactive, but they're still wheat. FODMAPs and wheat sensitivities. And so this is from Ziegler et al. In 2016. And we can see that now with the tetraploids, durum and Emma, that at least related to bread wheat in the wheats that they chose, that there were less fructan or less total FODMAPs. And, you know, and this is repeated in the literature, although I've only got one paper, but I can't be doing a literature review on every single piece of this for you, is that this is equal to and arguably slightly higher than common hexaploid wheat in the FODMAP content. And then this is from another paper from Schmidt in 2021, and I've taken their data for rye, which on this scale was 3.3 grams per 100 grams. So the rye had a much higher level of the fructans. Now, like I said, and I'm going to go back and reflect on the value of rye in terms of health, is that for probably 90% of the population, these FODMAPs are not only they're not neutral, they're actually beneficial because they're beneficial for our gut microbes. Okay, is it the processing? And so I've just taken one panel out of the Ziegler et al. Paper. This figure has another one that's parallel to this, and so it's effectively the same results for spelt. And I think there are some really interesting things to bring out in this. So what we're looking here is at FODMAP content. Here we have fructans, and here we have raffinose, the galacto-oligosaccharide. And what they've done, and they've called this proofing times, but I went and looked careful detail at their methods, and this is what we would probably call bulk fermentation times, because there was a 30-minute final proof after shaping. And what we see is that with a 1 hour fermentation, we get a considerable reduction in the amount of fructans, and we get a considerable reduction in the amount of raffinose, but we get this excess of fructose because the fructose is being cleaved off because fructans are a fructose polymer. And in this early part of fermentation, there's not enough time for the bacteria to reprocess this and use it for their own energy needs, or the yeasts. And so for people who have got irritable bowel syndrome and sensitive to FODMAPs, if 1 hour fermentation might actually be worse. But as we look now, I'm just going to focus on the 4.5 hour bulk fermentation, is that we've reduced the total fructans, the total FODMAPs by 90%. So this is really valuable. And this is repeated in the literature quite, I think, sufficiently for me to be a consensus. Just a note on rye breads and mannitol. Rye breads are high in fructans already. When these fructans are degraded, the fructose can go through a shunt that ends up producing a polyol called mannitol. And in this particular paper, they argued, and I know that Mayuka showed us some low FODMAP rye breads, is that they could not create a low FODMAP rye bread, either through fructan reduction or because there was too much mannitol. So that's kind of interesting. Gluten sensitivity, yes, I've got a clock on, Sean. I know that there's 1 minute to go. is it the processing? And this is where, and I still kind of am confused with this, because this is just the newest piece of data I could find, but it repeats other data in the literature that shows that long fermentations will degrade the gluten proteins, but this was much more focused on the gliadins. And it's really, really interesting. So this is the composition, if we go from left to right, and if you look at the relative heights, the relative abundance of these gliadins in the wheat flour that was used to make these breads. This is a no-time dough bread, so maybe a total of like 1 hour fermentation in the total bread making process. And you can see that there's a small reduction in the presence of the gliadins, but this is the one that's just blown me out of the water. Long fermented yeast breads and long fermented sourdough breads have almost completely degraded the gliadins. And so if you add the degradation of the FODMAPs and the degradation of these gliadins, the anecdotes of people that say, I can eat these long fermented breads and I don't have any tummy troubles, but I can't eat the supermarket bread, start to have more and more credence. And the only thing that surprises me about this is there are traditional wheat-based foods, pasta, couscous, bulgur, some of the breads of South Asia that are traditionally not fermented at all. So, you know, I'm still kind of at an aside from this. And so this is my last slide, Sean. I really would like to speak to it because this is where this has been popping up in my head more and more and more. So Alessio Fasano, who is a key researcher in the clinical side of gluten and wheat sensitivities, and this is a paper from 2011, and quoting it here, that regardless of, and I'm going to say regardless of the source of this, that changes in intestinal permeability preceded the activation of the adaptive immune response in celiac disease. And so we just heard from Ayuko Kulemonen about gut microbiota and gut barriers and short-chain fatty acids. And I'm going to focus on just real particular points here from this Mackie et al. Paper. A Western diet, which is low in dietary fiber, has very, very low production of short-chain fatty acids, and even some branch-chain ones which are detrimental. We get poor epithelial health, poor mucus production. We get toxins and peptides and bacteria in touch with the epithelium that shouldn't be there. The tight junctions are downregulated, so the gut is already leaky. If we go to a high fiber diet, lots of short chain fatty acid production, healthy mucus that keeps peptides and toxins and bacteria away from the epithelium's healthy, and it has tight, non-leaky junctions. So I'm going to argue, my hypothesis is, and I'll leave you this question, is that in a population that has chronically eaten a low fiber diet for at least 100 years, is the gut already leaky, and is that some of the causes of the uptick in wheat-related sensitivities? Thank you and good night.